Morning everyone. So today we are going to continue with the heredity and evolution chapter. I have already started the chapter and we have already discussed what is inheritance, what is heredity, what are inherited traits and all these things. Today we are going to start the next topic, which is rules of inheritance of traits, and these rules of inheritance of traits were framed by Mendel. So we are going to start Mendel's contribution in giving the rules of inheritance of traits. Mendel used a lot of contrasting characters for his experiments to prove out or give out the rules of inheritance, and for that he used a plant whose name was a pea plant, the common pea plant or Pisum sativum. So he used pea plant for his experiment for a major reason, that it produces a number of contrasting traits, or it has a number of contrasting traits. So what is the meaning of contrasting traits? It has traits which completely show opposite characteristics. For example, tall and short are two contrasting traits of the character height. At the same time, there are two other characters. If I take, like, white and violet are two contrasting characters of the color of the flower. So like this, two contrast of the character color of the flower. So like this, Mendel has taken a number of contrasting characteristics or contrasting traits, and in pea plants there are around 7 pairs of such contrasting traits. That is why Mendel has done his experiments to prove the rules of inheritance on pea plants. First experiment done by Mendel, he has only taken one trait. First Mendel's experiment, he has taken only one trait, and that is why, or one character rather, and that is why it is known as the monohybrid cross. It was called as monohybrid, where mono means one, and he has taken only one character, and the name of the character that Mendel has taken was height of the stem. Height of the stem. Whose stem? It is the stem of Pisum sativum. So height of the stem. The traits, the contrasting traits of this character were tall height and dwarf height, or you can write short height. Now these traits, when they were expressed in the parents as capital T and small t. So the parent who was showing the phenotype as tall, the genotype of the parent was capital T, and the phenotype of the parent, if it is dwarf, then the genotype will be small t. Now you can see here, this is a pure homozygous traits which are taken. Homozygous means both the alleles of it is same. That is why it is called as a homozygous trait. Here also it is homozygous because both the alleles of the pair of factors are same. Now Mendel used the word factors for genes. So here the gene is stating height. The height trait is tall. So tall is represented by two homozygous allele, which is capital T, and the dwarf is small t. Then he has then taken the gametes out of it. We all know that when we are representing gametes, the number of chromosomes will become half. So here we are taking either of them. So capital T is one factor, or another factor will be capital T again. Here there are again will be two factors, so small t and small t. So these are the pairing. In the parents it was in pair, but when it was forming the gamete, each cell was receiving one of the two factors. Hence the gametes were formed like this. Now let us take one of the parent as a male. So let this be the male parent and this be the female parent's gametes. So this is the male gametes and this is the female gamete. Now I will be writing the gametes of male and female gamete in a in a square box, and the name of this square box is Punnett square. Now in this Punnett square, I'm writing the male gametes on one side, which was capital T, capital T. These were all male gametes according to the notifications that we have taken, and the other side we have taken is small t, small t. Now when we will be fertilizing them, means here male gamete is fusing with the female gamete, it is giving capital T small t, capital T small t, capital T small t, and capital T small t. As you can see, all the possible offsprings of this is a female offsprings are all tall, all are capital T small t, and though it was genotypically capital T small t, but phenotypically it was representing tall plant. It was representing tall plant, and of course it was a cross pollination. So during the cross pollination that Mendel has done, the offspring which was produced, this was called as the F1 offspring or first filial offspring. In the first generation, all the plants phenotypically or physically, all the plants were tall. Mendel was not able to identify what is the genetic difference because he has no idea about that. There was no electron microscope at that time, so he was confused that when I have given a tall parent along with a dwarf parent, then where does the dwarf character go? That is the first problem. And the second problem was, why was there no moderate or no halfway characteristic shown in the F1 generation? So he thought of doing a cross pollination. This cross pollination offspring was taken for self pollination. Now the idea of self pollination came to the fact that if this tall plant is same as that of the parental tall, then all the offsprings will become tall. But if this F1 plant is not same as the parental tall, then there will be different in the offsprings, difference in the different types of offsprings will be produced. So he carried out the second generation cross, and now the second generation cross was a self cross or a self pollination. So in the self pollination, what he was doing, now let's see what he did in the self pollination. He had taken two varieties. Now he thought, he was having no idea that which plant, the plant which he has got in the F1 generation, it is a parental tall. This is a parental tall, homozygous tall, and this is a heterozygous tall. He has no idea seeing this on the field. When he saw the plant in the field, he had no idea about that. So what he did, he cross pollinated. Now if I will cross pollinate F1, this one was the F1 generation hybrid and this was a parental one. So when I will self cross the F1 generation hybrid, this is the parent, the gamete formed will be capital T small t and capital T small t, and in the Punnett square where I will see, I can see that capital T small t, capital T small t, both the sides I am writing the gametes of both the organisms, one male and female. I will see that here the offsprings that are formed are like this, means there are capital T, there is a point, there is a capital T small t, and there is also a point of small t. Now capital T is tall, capital T small t is also tall, while 1/4, that is capital small t is dwarf. So he got two types of offsprings here. 3/4 are tall, while 1/4 is short or dwarf. While if it would have been same as that of the parental tall, then let's see what would be the difference. So the difference will be like this. If I go on putting self pollination of the parental type, I would have got this type of offspring in which all offsprings, just take a guess, all offsprings are tall here. So this is the F2 offspring. So F2 offsprings here all are tall because all are capital T. But here in this case, it is not this capital T small t which we have got in the F1 generation by crossing homozygous tall and homozygous dwarf is not same as the parental tall. It is a different type of tall, and this, because of this tallness, we have got a dwarf character after self pollination. As you can see here, 1/4 offsprings are dwarf. So from this, Mendel came to a conclusion. Now, what is the conclusion that Mendel arrived at? Conclusion that Mendel arrived at is, as the F1 generation is tall plant, which is shown by like this, and Mendel didn't know what was the genotype. Mendel was only having the idea of the phenotype tall. He confirmed that this offspring which has come in the F1 generation is heterozygous and is not same as the parent. It is, this tall plant is not same as the parental tall. This is not same as the parental tall. The F1 tall is not same as the parental tall. And second thing which he got to know from the conclusion is that the offspring of the F1 generation, it has both the alleles, that is tall and dwarf. So F1 generation offspring or hybrid has both tall and dwarf alleles, and because it is having both the tall and dwarf alleles, therefore it is able to represent, it is able to show off the characteristics of dwarf in the F2 generation, as it was showing, it was showing the dwarf trait in F2 generation. So this was the conclusion that Mendel got. Now after this, Mendel had the inference. Out of it, Mendel took out the inference of these observations and conclusions. From this, the inference that Mendel got was that the tall trait was dominating. The tall trait was dominating the dwarf trait. The tall trait was dominating the dwarf trait, hence only tall trait was available, was seen in the F1 generation. But when it was when self pollination was carried out or selfing was carried out, at that point of time, the dwarf trait was visible in the F2 generation, but only in the homozygous condition. From this, he gave the first law of inheritance, which was law of dominance, and according to the law of dominance, one trait suppresses the expression of the other trait. One trait suppresses the expression of the other trait. So in this particular experiment, the tall trait is suppressing the expression of the dwarf trait, and dwarf trait is not able to express in the F1 generation and is only able to express itself in the F2 generation after selfing, and that too only in homozygous condition. So this is all about the law of dominance, which was taken as the first rule of inheritance, which is called as the law of dominance. Thank you everyone for your patient listening. I hope tomorrow when I will come upon with a new video, I'll be telling about the other types of laws of inheritance or other rules of inheritance given by Mendel. Thank you everyone.